Welcome to today's Masterclass Explainer class. We are jumping right into the incredibly dynamic, absolutely crucial molecular machinery that sustains, well, pretty much all life on Earth. I'm super thrilled you're tuning in because today we're tackling photosynthesis. Now, I know it's a topic that can seem pretty dense at first glance, right? But I promise you, once you see how these microscopic pieces fit together on such a grand scale, it is literally mind-blowing. We're going to break down these complex light reactions into really simple, clear steps to make absolutely sure you understand exactly how light energy transforms into chemical energy.
Okay, so here's our game plan for this session. We'll hit unlocking the photosynthesis mystery, pigments and paper chromatography, chloroplast anatomy, factory floor, light harvesting and wavelengths, the Z-scheme and splitting water, and finally wrap up with biology exam essential takeaways. We're zooming all the way from the visible leaf down into the subatomic electron transport chain.
All right, section 1, unlocking the photosynthesis mystery. Let's start with the early experiments that kicked this whole thing off.
So, where do plants actually get their food? I mean, have you ever stopped to wonder how an autotroph survives when it just can't hunt or forage like we do? We know all animals, humans included, completely rely on plants for food at the end of the day. But plants, they have to make or synthesize their own food. They use this fascinating physicochemical process to literally grab light energy and use it to build organic compounds. But how did we actually figure out what goes into this process?
Well, early experiments gave us the answers step by step. There's this classic setup where you take part of a plant leaf and stick it inside a test tube containing cotton soaked in potassium hydroxide, or KOH. Now, the absolutely crucial detail here is that KOH absorbs carbon dioxide. The other half of that exact same leaf is left outside, totally exposed to the air and the sunlight. Later on, you test both halves of the leaf for starch, which, by the way, is exactly how plants store their synthesized glucose. The exposed part tests positive, but the half locked in the tube without CO2, it tests completely negative. This brilliantly proves that you absolutely must have both sunlight and carbon dioxide for a plant to make its own food.
Moving on to section 2, pigments and paper chromatography. Let's figure out what's actually catching that sunlight.
If you've ever stared at a tree and wondered why there are so many different shades of green, the secret is in the pigments. If we run a paper chromatography experiment, and hey, put a massive star next to this in your notes because it is highly testable, we can separate the leaf's color into its specific chemical parts. First, you just extract the leaf pigments. Next, you apply that extract right onto your chromatography paper. Finally, you just sit back and observe the separation by color as the solvent carries them up the paper based on their unique chemical properties.
What this shows us is that a leaf's color isn't just one single pigment. No way. It's a whole team of distinct molecules working together. On the chromatogram, chlorophyll A shows up as this bright or blue-green color. Chlorophyll B is more of a yellow-green. Then you have xanthophylls, which are pure yellow, and carotenoids stretching from yellow to a yellow-orange. Now, they all chip in, but chlorophyll A is the boss. It's the absolute chief pigment for photosynthesis. The others are accessory pigments. They essentially widen the net to absorb more types of light, and, this is super cool, they actually protect our VIP, chlorophyll A, from photooxidation. All right, section 3, chloroplast anatomy, factory floor. Time to shrink way down and step inside the cell. Behold the chloroplast. I want you to think of this incredible little organelle as a super efficient, highly organized factory floor. Looking at a cross-section under an electron microscope, you immediately spot a complex membrane system. You've got the grana, which honestly look just like tiny stacks of coins, connected by stroma lamellae. And all of this is floating in a fluid matrix that we call the stroma. And in this factory, there's a very strict division of labor. Seriously, everyone has a specific job. The membranous grana, they are entirely responsible for trapping light energy and pumping out the high-energy molecules ATP and NADPH. We call these the light-driven photochemical reactions. Meanwhile, the fluid matrix, the stroma, handles the enzymatic reactions. You might hear these called the dark reactions or carbon reactions. The stroma's job is to take that newly minted ATP and NADPH and use them to physically synthesize the actual sugars. Section 4. Light harvesting and wavelengths. Let's look closer at those pigments catching the light. Now, this is where it gets really elegant. There is a perfect overlap between absorption and action. If you map out the absorption spectrum of chlorophyll A, meaning you track which specific wavelengths of light it absorbs best, you get these massive peaks in the blue and red regions of the visible spectrum. And when you overlay the action spectrum of photosynthesis, which is just tracking the actual rate of photosynthesis at those different wavelengths, they match up almost perfectly. The absolute highest rates of photosynthesis happen right exactly at the blue and red wavelengths, because that's precisely where chlorophyll A is soaking up the most light.
Deep inside the chloroplast membranes, these pigments are grouped into two discrete light-harvesting complexes, photosystem 1 and photosystem 2. For your exams, you absolutely have to know the difference between them. The reaction center for photosystem 1 is called P700 because its central chlorophyll A molecule absorbs light best at exactly 700 nanometers. Photosystem 2's reaction center? That's P680, hitting its peak at 680 nanometers. Oh, and a quick pro tip for test day: don't let the numbers fool you. They are named strictly in the order they were discovered, not the sequence they actually function in.
Section 5, Z-scheme and splitting water. Get ready for the main event, subatomic electron transport.
Okay, before any electrons can actually move, they need a serious jolt of energy. Enter the light harvesting complex. Think of it like a massive, highly tuned satellite dish. It's built from hundreds of those accessory antenna pigment molecules we talked about, all bound to proteins. They act as this incredible antenna array, gathering incoming photons of light and funneling all that trapped energy directly into one single, specific chlorophyll A reaction center. The goal? To massively excite its electrons.
And that brings us to the infamous Z-scheme. The best way to understand this is to visualize a roller coaster on an oxidation reduction scale. First, light smashes into photosystem 2. The electrons get so excited they literally jump rapidly uphill to a primary acceptor. From there, they take a smooth ride downhill through an electron transport chain made of cytochromes. Now, they aren't consumed here. They're just passed along the track to photosystem 1. At the exact same time, photosystem 1 is getting hit by light 2, which excites those electrons to jump rapidly uphill once again to a brand new acceptor. Finally, they drop down one last time to reduce NADP+ into energy-rich NADPH. It is a brilliant, continuous, non-cyclic flow of pure energy. But wait, let's think about this for a second. If photosystem 2 is constantly launching electrons up this roller coaster, it's going to run out eventually, right? To keep the ride going, photosystem 2 gets desperate to replace those lost electrons. It solves this problem by physically ripping apart water molecules. It's this dramatic process called photolysis. Imagine a water molecule being violently split wide open. It surrenders 4 hydrogen protons, it gives up 4 electrons to feed the system, and incredibly, crucially, it casts off oxygen. That's right, this microscopic subatomic event releases the exact oxygen that you, me, and every other animal on Earth breathes to survive.
Finally, Section 6, Biology Exam Essential Takeaways. Let's lock in the stuff you absolutely need to know.
Class, if you want to ace this material, you need to memorize these points right here. This is the absolute core of the light reactions. Fact 1, the primary pigment is always chlorophyll A. Fact 2, lock in those absorption peaks. PS2 is P680, PSI is P700. Fact 3, the splitting of water, photolysis, the very thing that gives us our oxygen, is strictly 100% associated with photosystem 2. Fact 4, the Z-scheme is a non-cyclic flow of electrons. And finally, remember the whole big picture. The entire biological purpose of these light reactions is just to bank high-energy ATP and NADPH, with oxygen being released as a brilliantly fortunate byproduct.
So there you have it. You now know exactly how a plant captures sunlight and turns it into cellular energy batteries. But as we wrap up this explainer, I want to leave you with a massive question to chew on before your next study session. How exactly does the plant take this newly banked ATP and NADPH energy into the stroma, pull completely invisible carbon dioxide out of thin air, and physically build the solid sugars that feed the entire world? Just think about that for a second. Keep that curiosity alive, keep studying, and I will see you in the next explainer.